All right, so our next lecture is about picornaviruses, and the most famous picornavirus is going to be the one that causes poliomyelitis. So, this is what we would call polio. And polio causes a lot of things. What it does is it damages nerves that go to a particular part of the body. And if this happens when a person is still growing, then you'll end up with really sort of weakened and often quite small limbs. If it happens that the picornavirus hits one of the nerves that controls your heart or your lungs, for example, then in that case it can lead to death. Or when it hits the lungs in particular, there used to be a machine called an iron lung that would just physically move your chest up and down doing compressions. And that can keep people alive. They can live for years and years on those. There's still some old polio sufferers who are in those, but there are not many left in existence of the iron lungs. So, shown here on the right, you got a little girl who has had polio, and you can see that she's got one leg that's a little bit smaller than the other and doesn't quite work right. The parts don't quite work right. And yeah, this is one of the things that can happen. You just get a really weakened small limb on one side because you don't have the nerves don't run it quite as well. In some cases, it can lead to paralysis of the hind limbs, all four limbs, yeah, all kinds of things. Shown over on the left side, this is an ancient Egyptian little funerary plaque, and this is a guy with by name of Roma, by the look of it, and he's got a similar sort of thing going on there. So there are a couple of conditions that can damage the nerves and lead to something like this, but this is referred to as the poliovirus plaque, if you like. And yeah, it's thought to be the first representation of this. So maybe, hard to tell. But if we could find the body, there are things that could be done, possibly testing, looking for little bits of the virus in there. Okay, so what is a picornavirus? Let's break it down. So, pico means small, RNA means RNA, and virus is virus. So, there we go. We just broke it down. Picornavirus is like the common name for these things. They actually belong to the family Picornaviridae, and virus families all end in the word viridae with something else in front of it. And these are grouped together. The Picornaviridae and several other families of Picorna-like viruses are grouped up in the Picornavirales. So that's the order there. Pardon me. Okay, these things are usually tiny, as the name would suggest, 25 nanometers in diameter. Compare that to something like a coronavirus, that's going to be more like 80 nanometers in diameter. And flu virus is going to be roughly 80 nanometers by, up to, I don't know, 4,000 nanometers if it's really long in diameter. And so, yeah, picornaviruses are about as small as viruses come. Um, Generally, they do not have an envelope on the outside, so they're going to have a protein coat and no lipid normally, but there's one, which is the hepatitis A virus, and that's shown up in those images at the top. The one that says V, you can see hepatitis A virus all on its own, and in the lowercase I and II, or 1 and 2, you can see some of these viruses that seem to be stuck to vesicles. So hepatitis A virus has the ability to stick to little pieces of membrane, and it's not really an envelope because it's not filled with virus proteins, but it is kind of wrapping itself in a membrane in order to hide. So yeah, clever little virus, kind of neat. Okay, so the inside of the virion contains a genome, and it's protected by a very rigid protein capsid that just makes a perfect little thing, kind of looks like a soccer ball. And there you can see it down there. It's made of a shell of three proteins called VP1, VP2, VP3. VP1, 2, and 3, the VP stands for virion protein. So, yeah, wow. And in the image at the bottom, around the center of the page, you can see that there is a fourth member of that group, which is VP4. And VP4 forms like a plug. It sort of sits on the back of these other things. And it's blocking the opening so that nothing can leak out of the virion until the right time. So you're going to watch VP4, and VP4 is going to get out of the way for sure. All right, so we're wandering a little further afield. So we've been dealing with envelope viruses exclusively so far. And we've done an envelope virus that has an RNA genome that turns to DNA, so that's HIV, and an envelope virus that has an RNA genome that stays as RNA, but it's positive strand when it comes in, so that's corona. And we've got a segmented negative strand RNA genome, that was influenza. And so with Picornaviruses, what we get are another slight variation on this. So we have a single-stranded positive sense RNA genome. So that right there where it says one strand plus ssRNA, that's exactly what that means. Single-stranded positive sense RNA genome. But the neat thing is that the other viruses we looked at had a special feature. Rather, influenza virus messenger RNAs and coronavirus genome had a cap and a poly A tail, and those two features allowed them to be translated directly. Now, picornaviruses also want to be translated directly, but they do not have a cap, which is kind of neat. They do have a poly A tail, so that's fine. That part works as normal. But yeah, in place of a cap, they have a small protein called VPG, which stands for virion protein genome associated. So it's kind of backwards from the way you would say it, but yeah, virion protein genome associated, VPG. And that's going to be really important. We're going to try and solve the mystery of how we get VPG onto the genome. That's going to be, I think, an interesting test question and thing just to build up to. But we'll get there. Yeah, right now we're just doing basics. All right, here are some famous picornaviruses. So at the top we got poliovirus. It mostly is a diarrhea virus in most people, but occasionally it'll get out of the intestine. It'll find its way into the bloodstream. And from the bloodstream, it'll find its way to a nerve cell. It'll track up that nerve cell into the spinal cord and track up that spinal cord into the brain. And if it gets to the brain, it can kill you. If it gets to the spinal cord, it can end up blowing up all the nerves that connect to some part of the body, which can lead to paralysis of certain parts. Yeah, so polio was really bad, and this was one of the first really successful vaccines. When this vaccine came out, they had people lining up around the block to take it. This was long before anti-vaxxers were a thing. Next virus on there is Coxsackie virus B3, named after Coxsackie, New York, if I remember correctly. And this causes an enlarged heart. And if you're a fan of the Grinch movies, you would say, oh, isn't that really nice? An enlarged heart. But the truth is that no, it is terrible because that enlarged heart is just sort of full of fluid and it's kind of weak and it doesn't do a good job of pushing blood around, which is the only reason the heart is there. The other viruses, and these are all in the genus Enterovirus, are human rhinoviruses, and they cause about half to two-thirds of common colds. The other one that we talked about was coronavirus, and those cause somewhere around 10-20% of common colds. And then there are about 40 viruses total that cause some version of the common cold. And so when people say, why haven't they cured the common cold? Now that you're in virology class, you can say, oh, cool, which one? Because, 40 different viruses takes at least 40 different solutions and real tough. Other important viruses, hepatitis A virus will cause hepatitis, so sort of a swelling and immune attack in your liver. But you can get sort of severe hepatitis in adults. You can end up sort of turning yellow, kind of like jaundice for a little while. Usually this passes without too much incident. And foot and mouth disease is something they really worry about over in Europe in particular. If they get the tiniest outbreak there, they will collect up all the cows or chickens or whatever from an area and just burn them. Giant piles of burning animals. That's what foot and mouth disease looks like to me. Over here in America, I think we just decide to live with foot and mouth disease, which causes terrible sores on the animals, and sometimes a little worse. But we like our mutant cattle, and we say that's okay, I guess. Okay, so we're going to overview the replication cycle, and then most of this set of lectures is going to be going through different parts of the replication cycle. This is a nice little thing that you can refer back to if you need it. So yeah, VP1, I like to remember it this way. VP1 starts the fun. So VP1 binds to the cellular receptor protein. And depending which picornavirus it is, it'll be a different protein. And we got a list of them coming up on the next slide. Then VP4, which is at the back of those VP1, 2, and 3, VP4 is the door. That's how I think of it. So it's like a little plug that is blocking the hole in VP1. The VP1s come together and they actually make a little 5-pointed star on the surface. And they can pull apart just a little bit and they can let that RNA come out of the virion and go into the cell. But VP4 is there to block the way until just the right time. And so there's an activation. It's going to be low pH. It's going to be after endocytosis. So something like going through a clathrin-coated pit, just like every single one that we've talked about, right? So yeah, once you learn one of these things, you got it. Then, furthermore, once we get into the cell, the viral RNA will be translated to make protein. And picornaviruses generally are going to make one great big protein. There are a couple of them that make two smaller proteins, each of which is half of the big protein. Either way, these are called polyproteins. So they consist of a lot of different proteins in a chain. And this is the same strategy that coronavirus did, with those 16 proteins that start out as one big long thing and are cut into little pieces by a protease. And that's the same thing here. So the big long polyprotein is made and it's cut into little parts by the protease. And the protease is going to be called 3C, but we will get into that. You bet we will. Okay, so that's step 4 and 5, then step 6. So we're going to make membrane-enclosed replication factories in the cytoplasm using some of the other genes of the, proteins from the picornavirus. And then we'll take that positive strand genome, copy it into a negative strand RNA template, and then copy that back to make more positive strand RNA to package up and send out. And each of the positive strand RNAs could be translated to make more protein or can be packaged up to go into a new virion. Usually in the process of translation, we're going to cut off that VPG, and we kind of need that VPG for packaging into the virion. So it's like a one or the other kind of deal. But the product looks the same. You can take one RNA and either turn it into protein or wrap it up in a new virion and send it out. And then the virion is going to be made from all the proteins from what we call the P1 region. So it's right up at the front of poliovirus. And so it's VP1, VP2, VP3, and VP4. VP1 makes those little 5-pointed stars. VP2 and 3 sort of fill in all the space around those stars and join things together. And then VP4 is the door, and it sits right on the back of VP1 and, just blocks the way so that nothing can leak out at the wrong time. And we will assemble that virion either around the RNA or it's thought that we may assemble the virion and then load the RNA in with a different protein. That's a theory that has not yet been proven for picornavirus, but I think we're going to operate as if that is a pretty good idea of what's going on. Okay, and so once we have assembled, then the virus, instead of like going through the Golgi apparatus or anything like that, just fills up the cell and then bursts it open. Just boom, like a balloon, like a dirty garbage bag. And then all the little thousands of picornaviruses can run loose and cause some other stuff. So these are really, really severe little viruses in terms of what they do to the cell. They completely take it over. They shut everything down in the cell. They just absolutely dominate the cell and then explode it. And this is the thing that causes the common cold that we're not so worried about. So it's kind of amazing. Really severe on a cell-by-cell basis, but overall, we just control it and move on. Okay, here are some names of receptors for picornaviruses. So these are good test question answer things. So let's see. We've got at the top, we've got ICAM1. And so I wrote out what each of these things stands for. It's the intracellular IC adhesion molecule AM number 1, ICAM1, ICAM1. And this is a protein with some immunoglobulin-like domains. So immunoglobulin-like domains are what you would find in an antibody. An antibody is made of several immunoglobulin-like domains. It's just a particular way that protein folds up. And yeah, a lot of picornaviruses, as you go down the list, you can see a lot of picornaviruses do tend to bind to those immunoglobulin-like domain proteins. Yeah, I don't know. It may be a shared evolutionary sort of tick from way back in the day. So human rhinovirus 10 can bind the low-density lipoprotein receptor. So this is the one that binds the, let's see, bad cholesterol, quote unquote, even though they're really talking about the low-density lipoprotein. You've got CD155, which is the receptor for the poliovirus. You got ICAM1 again, which is the receptor for not just human rhinovirus, but also Coxsackie A virus. You've got the alpha-2 integrin. So integrin is going to be involved in immune cell signaling. And that is going to be the echovirus, one of the echovirus receptors. And echovirus 2 by no, 2 echoviruses bind that. 6 echoviruses bind the decay accelerating factor DAF, and encephalomyocarditis virus EMCV binds to VCAM1. And you know what? We could keep going on this list. There are a lot of picornaviruses out there, like really a lot. Yeah, each one seems to have its own distinct receptor. So this is a good little overview. It shows you that all these proteins are rather, here, let's go look at these. So here are pictures of all these picornavirus receptor proteins. What do they have in common? Well, all right. Everywhere that you see a little loop that's shaped like an omega, I guess, all these things that look like a little circle that is closed up on both sides. Those are going to be the immunoglobulin-like domains. And so you can see we've got 1, 2, 3, 4 at the left, and then 2 proteins over. We got another one of those as the 4, 5, 6, 7th one along from the left. So a lot of immunoglobulin-like domains, but these are really common on the surface of cells. We also have proteins that are low-density lipoprotein receptor and SCR-like proteins. So that's the CD155, that should be. And integrin proteins shown there are a 2 subunit. They have an alpha and a beta subunit. And yeah, those are things that the some picornaviruses can bind. So what they have in common is that these are all proteins that are in the plasma membrane, and they have a domain that sticks out of the plasma membrane, and that's where the virus is coming along and going to bounce into it and hopefully find a cell so it can replicate. So all it takes is for the virus to be able to grab onto something. And viruses are constantly sort of mutating, evolving, and if they just happen to land in a place where they can make a decent contact with some new receptor, then they will change and they'll bind to a different receptor. And you know what? If it gets them in and it gets them replicating, then that is good enough. All right, that's been part 1.